Bueno, pues buenos días a todos. And now I will switch to English to introduce Professor Jean-Pierre Sauvage. First, on behalf of the Dean of the Faculty of Chemistry, Professor Adela Maury, the organizing committee of the 125th anniversary of the studies of chemistry at the University of València, and of all the staff of the faculty, I would like to thank Professor Jean-Pierre Sauvage for accepting to give this lecture, unfortunately still online. However, we hope to have you shortly with us, probably in October. To introduce Professor Sauvage is very easy because he is one of the biggest names in chemistry worldwide. Just to remark a few points of his trajectory, I would say that he performed his PhD studies at the University Louis Pasteur of Strasbourg under the supervision of Professor Jean-Marie Lehn, Nobel Prize in Chemistry in 1987. During his PhD work, Jean-Pierre Sauvage contributed to the preparation and study of the first cryptons, macro-bicyclic receptors able to encapsulate selectively alkali and alkaline earth metal ions. Then he made a postdoctoral stay at the University of Oxford with Professor Malcom Green, working in transition metal chemistry and organometallic chemistry. He returned to Strasbourg, where he was promoted to research director in the CNRAs, equivalent to our CSIC, and since then his group has been working in different topics, most of them joining organic chemistry with transition metal coordination chemistry. One of these topics contemplated the combined use of organic ligands and the ability of metal ions to organize the space through coordinative bonds to build amazing chemical structures and systems. Starting by two interlocked links, he moved towards the design, preparation, and study of molecular devices able to perform oriented motions in response to chemical, electrochemical, or photochemical inputs, namely molecular machines. All this amazing chemistry was acknowledged in 2016 with the Nobel Prize of Chemistry that he shared with Professor Fraser Stoddart and Ben Feringa. Jean-Pierre, thanks again for your availability to give this online lecture and for having hosted in your laboratory many PhD students and postdocs coming from the University of València. I think this online audience is very much looking forward to attending your lecture. Thanks for all, Jean-Pierre.
Thank you, Enrique. Thank you. I'm a bit disappointed because I do not see any face, on my screen. I just see my lecture. And this lecture, I already know it pretty well. But no face, no photos. But okay, so let me start by telling you that I'm very happy to be in Valencia. Honestly, this is my favorite city in Spain, also because I have several very good friends there, including Enrique and others, and a real pleasure. So we will talk about topology and molecular machines today, and I will show you that those two research fields are very closely interlinked or intertwined. And this is the philosophy of today's lecture. So we have to talk about a few things before, some definitions, and in particular topology. So topology, to make it very simple, this is the science of infinitely deformable objects. So you can distort the objects, and the topology of those species is not going to be modified. It will be the same. Chemical topology is very similar to topology in general. You can, by your mind, of course, only by your mind, pull on bonds, so elongate bonds, shrink bonds, and totally distort angles, provided you do not cleave anything. The topology of the species the molecular topology of the compound will still remain exactly the same. So many years ago, I think mathematicians were interested in knot theory, which is a part of topology, it's a branch of topology. And here are drawn a few knots. They are prime knots because you cannot divide them into other knots. And you see that the simplest species here is the trefoil knot. So the trefoil knot, it's very beautiful, and many people have dreamt of this species. And in our group, we have also been very interested in knots. And you should know that today, mathematicians have listed something like billions of knots, much more complex. Everything started in chemistry with a very important paper published 60 years ago, or even 61 years ago, by Frisch and Wasserman. And they published in JACS a review or a discussion. At the time, there was no experimental work, but it was the first publication mixing the two words, chemistry or chemical and topology. So it was bringing together two fields which were very remote from one another. And so those two guys, Frisch and Wasserman, they were working for a chemical, or not a chemical, a big industry, Bell Telephone Laboratories. They were doing their research there. And at the time, I think it is remarkable that big companies didn't care about having researchers paid by them, by the company, doing basically whatever they like to do. And so those two persons were crazy about topology. They were experts in topology, and they brought this field into the chemistry world. Very important. And so there are a few definitions to know. If you have a ring, which you can distort, of course, you can make it an ellipse, a square, a triangle, what you like. The topology will still be the same, as I said. But if you can draw it in a plane, like a ring, you will say that its graph is planar. So this species has a planar graph, and thus the topology of it is said to be trivial, no interest. On the other hand, if you take exactly the same sequence of atoms and chemical bonds, you can arrange it in a very different way. You can make a knot, a trefoil knot, and this time, this trefoil knot has a non-planar graph, because if you want to project it in a plane, draw it on your sheet of paper, you will have to introduce 3 crossings. And thus, topologists would say that this molecule is non-trivial in terms of its topological properties. And at the time, of course, there were very few non-trivial molecules from a topological viewpoint. And so we have to start somewhere. And there were discussions many years ago, even at the very beginning of the 20th century, probably in Zurich and maybe in Germany, on catenanes. So you all heard of catenanes nowadays. The simplest catenane is this one. It's a 2-catenane because it consists of 2 interlocking rings. The word catenane comes from Latin. Catena in Latin means chain. Maybe the same in Spanish, I'm not sure. It is the same in Italian, in a way. Catena means chain. And this is indeed the smallest chain you can think of, because it consists of 2 cyclic links. Rotaxanes have always been associated to catenanes, but strictly speaking, they are not topologically interesting species. They are trivial. Now, as I said before, many chemists have been interested in catenanes, but mostly from an intellectual viewpoint. And I think the first fully convincing piece of work published by chemists on catenane is due to those two great chemists, Gottfried Schill and Arthur Lüttringhaus. So Lüttringhaus was kind of a legendary organic chemist in Germany, and Schill was his student, and they published a beautiful paper in 1964 on the making of a catenane. As I said before, it was the first very clear paper, very convincing paper on a catenane. Beautiful work that everybody, I think, greeted. Well, I was too young, to be interested in that. But there was a problem, or let's say a difficulty, which was that it was a very long procedure. It was a 23-step synthesis for making a catenane. And so with such a difficult strategy, very few people were interested in catenane. Nobody really tried to repeat the work, and very, very few people started to do work in this field. It appeared to be too difficult. So we had to wait for some time, and I will know how we entered the field and how we made catenanes. So when I had been working with my boss at the time, after I came back from my postdoc in Oxford, with Jean-Marie Lehn, we started a new project in the group. We were very excited about trying to split the water molecule to H2 and O2. And it was a bit after the energy crisis, the oil crisis in the 70s. And so we could produce some nice work, I believe, which was the first photochemical reduction of water to H2. And so at the time, the main hero was a ruthenium complex, so this species. And ruthenium complex, this ruthenium complex, ruthenium traced by pyridine, was extensively used by photochemists and photophysicists in relation to water splitting, but also for its photochemical properties. And there were really hundreds of papers published every year, or perhaps even more, on ruthenium traced by pyridine. The difficulty with ruthenium traced by pyridine lies in the price of ruthenium. You know that ruthenium is a noble metal, very expensive, similar to rhodium or platinum. And so we thought that maybe we should try to replace ruthenium for a much cheaper metal. And David Macmillan, a real pioneer in this field, had worked on copper, and he could show that some copper complexes can be very interesting from their photochemical viewpoint. And so copper compounds were very promising. It happened, and I think it was not completely an accident, but it was a very happy event, that Dave Macmillan took his sabbatical leave in Strasbourg, and that was at the beginning of the 80s. And of course, when he arrived in Strasbourg, he communicated with us because we knew each other from the literature, and he visited our group, and then we started to collaborate. And at the time, we had made a molecule, a very simple molecule, simple looking at least, but it was a new molecule. And we made it for various reasons in relation to catalysis, molecular catalysis, using iridium complexes, and also for the photochemical properties. And so with Dave, we decided to collaborate immediately, and we made the copper complex here. And later on, David Macmillan and his group studied all the photochemical properties, all the photophysics, and it was a very pleasant and very fruitful collaboration. But there was something else. And at the time, it was before ChemDraw, and all the softwares which are today available. And we were drawing the molecules by hand, ourselves. And I was in charge of drawing the molecules using Indian ink. Very difficult task. I was not so good, but I became better. And so that was the drawing I produced. And if you look at the drawing, you can immediately that there is something very promising potentially. If you materialize the two endpoints of this ligand here, this one lying in a vertical plane, and separately, if you materialize the two endpoints of the other ligands, this phenanthroline here, lying in a horizontal plane, you can notice something really very exciting. Now, if you interconnect the two blue points and separately the two red points, you make a catenane. If you look, it's very obvious. You have a ring here which contains the blue fragment, and you have another ring here incorporating the red fragment. And those two rings are interlocked forever. And so we decided that it was a very exciting observation, and maybe we should jump from water splitting, small molecules, activation, and catalysis into a new field. It was almost a virgin field, the field of catenanes. And this is what we did. At the same time, and it was also a very happy event, a very good friend of mine who was a great organic chemist, Christian Dietrich Buschecker, who very sadly passed away some time ago, decided to join my group, and we started to collaborate. And she was a fantastic chemist, as I said, a great friend and a great organic chemist. And within a couple of months, she succeeded. So she made the first catenane, which can be made practically, just in a few steps. And we published our first paper within 10 months after she had started the project. So the principle, again, I will show you. We have two strategies. The first strategy, we called it the entwining strategy, entwining like that. So we start from such a compound, which is very easy to make. It's a two-step synthesis from phenanthroline, the commercially available compound. And if we mix with copper, we generate the entwined species. Copper is at the center. And now we can cyclize on the right and on the left. It's everything. It's a one-pot reaction. And we obtained our first catenane. The yield was not super good. It was between 25 and 30%, but it was very acceptable. And in fact, historically, the first strategy we tried is this one, which we call strategy B. It's a little bit more complex because we have to make a ring first. It's a 30-membered ring. It's a two-step procedure again. And now we mix with copper, with another phenanthroline, and finally we cyclize. And this strategy is certainly more interesting in terms of making sophisticated molecules than the first one, because we can synthesize asymmetric species. So just to go on, we could crystallize the compounds, and the X-ray structures are shown here. We could demethylate using potassium cyanide. We can remethylate, and these two reactions are quantitative and instantaneous at room temperature. And maybe you can note that the two structures are very different from one another. Here, we have a very globular structure, and here we have an open structure for which one ring can glide almost freely within the other ring. And we call this conversion here a metamorphosis. And this is indeed a metamorphosis, a complete transformation. Let me pay homage to the people who made catenanes. And again, this is kind of history here. I mentioned the work of Wasserman, mostly a very interesting discussion on topology and chemistry, Schill and Lüttinghaus in '64. And then for 20 years, or almost 20 years, as I said, nothing happened. We started and published our first paper in '83. Later on, Fraser Stoddart and his group became interested in catenanes, and they joined the small club of catenanes. Chris Hunter, Fritz Föckle, they used hydrogen bonding templates. Fujita worked on palladium, also produced beautiful work. More recently, David Lee first published his original work, the beginning of his original work in 1995. And now they have done fantastic work, and they are still doing gorgeous work, probably inspiring lots of other people. And I would like also to thank Professor Ogino, who was the first to make very convincingly a rotaxane. Just before we go on, I would like to say that we have been much interested in topology. And before molecular machines, which started during the mid-90s or so, we devoted a lot of work, a lot of time on new topologies, and in particular on knots and on more complicated species, the Solomon links. And so we have been interested in that in parallel to the work on molecular machines. But we will not talk about that. Now, at the time, some people were asking us, so why are you making such molecules? They have no future. They are fun, of course, but they do not exist in nature. And that was wrong, because it was shown during the 80s and later the 90s that catenanes and knots are very, very common in biology. And we will meet them, literally, with DNA, with proteins, and even viruses. So if you look at what the people have shown in biology, molecular biology, this slide is particularly representative. The people could characterize knotted DNA, mostly from circular duplex DNA in plasmids. And so you have beautiful pictures here, photographs of knotted DNA. Of course, the scale is not the same as molecules, like what we understand by molecules, we as chemists. They are much larger. We are in the micron scale, but still they are molecular species. Some other examples, I think this one is particularly exciting. Whitehead was an English topology expert, a mathematician, and this very strange catenane, so to say, or link, using the topological vocabulary, was discussed. And DNA can also form Whitehead link, and this is shown here. It's a catenane with 5 crossings. So beautiful work. Proteins form knots. And this is something which surprised many people. You know that in Cambridge, there is a protein data bank, and all the structures, the X-ray structures of proteins solved all over the world are collected and gathered in this data bank. And some people explored this data bank, and they found that a little bit more than 1% of the proteins are knotted. Of course, it means that knotted proteins are relatively rare, but not exceptionally rare. There are really many knotted proteins in the world. And probably the most spectacular example is that of this bacterium, HK97. It's not dangerous. Don't be scared by my slide. It's a bacteriophage virus which invades bacteria. It will mix its DNA with the DNA of the bacteria which are invaded. It's not an RNA virus, contrary to the most famous of today's viruses. And if you look at the membrane, I think the beauty of it is that the membrane or the envelope is kind of a chainmail, because you have cyclic arrays of proteins here, those cyclic proteins, and they are all interlocking with one another. So you have something which looks like a chainmail. I don't know in Spanish, but in French you say côte de maille. And a chainmail allows this virus to be very flexible. It can distort very easily, but it is also mechanically very strong. It can pass barrier and find its way so as to invade bacteria. Now let's talk about molecular machines. And you will see in a minute that molecular machines are very tightly connected to catenanes or rotaxanes. So first, some very important statements. Synthetic molecules are most of the time considered as static objects. Although, of course, they can distort, they can vibrate, elongate. They normally, very, very few systems were set in motion on purpose. And this is why we say that they are static objects. Again, considering that there are stochastic movements. By contrast, now, if you look at biology, in biology, controlled motion is absolutely everywhere. It is absolutely essential. And we will see just two examples, but you should know that there are many motor proteins, which are proteins, sometimes very complex proteins, which are set in motion. And this motion has a function, very important function. And there are rotary or linear motors, walkers, systems which can contract or elongate. To make it short, they are everywhere in biology, in any living organism. Two very important examples. ATP synthase, so you know that ATP is the fuel of biology. Just to remind you, ATP, we use it as our energy source. When it is hydrolyzed to ADP, of course, you gain energy and you degrade ATP to ADP. And this is very important. You know, our human beings, our body converts ATP to ADP every day with, it's a very large scale. We convert about 1/2 of our weight of ATP to ADP. So you can calculate how many kilos of ATP you convert every day. And then we have to regenerate ATP. We do not throw away the degradation products. You know, we are very concerned about our surrounding, contrary to when we burn a fuel in an engine, in a car engine, where CO2 and water are thrown away simply. No, we recycle ADP. And the enzyme which is able to do that is ATPase. So ATP can be made from ATPase, we will say ATP synthase, but ATP can also be hydrolyzed to ADP, and of course it's not ATP synthase, it's ATPase. It's a very complex enzyme which has been studied in detail. It's a rotary motor. So you have here kind of a rotaxane, by the way, an axis threaded inside this big cluster of proteins. you have some kind of cavity here, and you have several cavities at the border between the green and the red proteins. And if you look carefully at the ADP now, which is yellow, and inorganic phosphate, which is blue, arrive, they insert themselves somewhere here in those slots, so to say. And then there is some chemical reaction taking place, and ADP is made and expelled. This is the purple species. Very, very efficient. Anything which lives on Earth is based on ATPase. So from most primitive living organisms to mammals and to human beings. Another very important enzyme is Nessim. And in the cell, something which is very important is to be able to carry molecules from a spot somewhere where they have been made in biosynthetic factories to where they are needed in the cell. And there is some kind of transporter, carrier, if you like, which is the kinesin, this little thing here, which walks on a big cylinder, which can be very, very long. It's a self-assembled cylinder, microtubule, and this cylinder allows the kinesin to walk on it, or even to run on it. And now the big thing you see at the back of the kinesin is a bag. It's a huge bag. It's a vesicle which carries all the molecules which have to be transported from dot A to B, proteins or hormones or pieces of DNA or RNA. So all these molecules have to be carried. And this is really a fantastic system. Just a quick point, this is not science fiction. You should know. Of course, the video is very spectacular, and you could believe that it's a bit similar to Star Wars or whatever, but it is based on many, many, many very rigorous studies, and our biologist friends know a lot on kinesin and the way it moves, the structure is known, and so this is not science fiction. Now let's ask ourselves the question, why are molecular machines important in relation to molecular topology? So let's now try to interlink those two things. It's very obvious. If you think of rataxanes or catenanes, and if you are able to set these systems in motion, you are on your way towards molecular machines. If you look a molecule of this type, and in particular Fraser Stoddart and his group have made beautiful systems of this type, and in our group, thanks to the work of Pablo, we have also made catenanes and rataxanes which move. So you are basically making a linear motor. You go from the left to the right, you come back to the left to the left, or to the left to the right, right to the left, and so this is a linear motor. And if you look at those two systems at the bottom, you are close to making a rotary motor, although directionality will be very difficult to introduce, but at least you make some oscillating machines or pirouetting machines. So now let me talk about the very first molecular machine we made in our group, and it's a pirouetting machine. So we start from here. It's a catenane. And this catenane is a bit similar to the one we have seen before, except that there is an additional fragment here at the back. We have 3 nitrogen atoms, and so this fragment is called the terpyridine. And I believe Enrique has done beautiful work on terpyridines and related ligands. And this terpyridine, of course, for the moment, if we have copper 1, does not interact with anything. Very simple. Copper 1 is a D10 electronic configuration metal, and basically it's not a real transition metal. So copper 1 is very happy with a surrounding of 4 nitrogen atoms disposed as a tetrahedron. This is what allowed us to make catenates. Now we will perturb the system. So we send a signal, we abstract an electron from copper 1 to copper 2. Copper 2 plus is D9, and copper 2 hates to be tetrahedrally coordinated. So copper 2 prefers to be 5 or 6 coordinated. And thus the system will react. The ring will glide here, will undergo a 180 degrees rotation so as to generate a new species. The terpyridine, which was originally here, will now replace the phenanthroline, and we obtain a very stable system with a very happy copper 2 because it is coordinated to 5 nitrogen atoms. Now we can go back, we reduce copper 2 to copper 1, we generate a very unstable copper 1 complex, which rearranges and we regenerate the starting form of the complex. So note something here. The two redox potentials are very, very different because here you oxidize a very stable copper 1 complex, so you have to apply a surprisingly positive potential. And once you have rearranged the system, copper 2 is very stable, so you have to apply a negative potential to reduce it to copper 1. So this system works fine, and we have a video which was made by a former student of mine, Sylvestre Bonnet. The video shows what I just said. We start from copper 1, tetrahedral. We abstract an electron, we generate copper 2. The system rearranges, we can inject an electron back, and it rearranges back. So it's a nice video, but it's also working the same way with the real molecules, and everything is super stable. We've never noticed any decomposition. Now let me talk about the gorgeous work of Ben Feringa and his group, which they published in 1999. And they were the first, or let's say they were the first to make a light-driven rotary motor. There were some people simultaneously who made a chemically driven rotary motor. Beautiful work, but very, very tedious. And in their case, it was very simple. So you take a double bond here, and probably you remember from your organic chemistry lessons that if you have a sterically hindered double bond, when you irradiate it using UV light, this double bond will photoisomerize. And so this is exactly what happens. Ben Feringa and his group, they were interested in photochemically driven switches, and they irradiate here, and the system photoisomerizes, and they obtain a new species. And then, if you wait for some time, or if you hint a bit, the system continues to rotate, and you generate a third form for the same molecule after some additional rotation. That was more or less found by accident, which doesn't withdraw anything from the merit of the group here. And they were very, very clever because they observed something which was new, which was kind of unexpected. And this led them to real rotary motors. From here, now you shine light again, and the system goes on. And now you have to heat a little bit to regenerate the starting form of the molecule. So basically, you have 4 states and 4 processes, 2 photochemical processes and 2 thermal processes. Now, let me show you the movie which they published on the internet. It's a beautiful movie, I think. Maybe you can see it, maybe you will have difficulties, I'm not sure. I told you there are 4 steps, 2 photochemical steps and 2 thermal steps. When you have a thermal step, you see that, the symbol, and you have a photochemical step, you see a big flash. Big flash, thermal step, big flash, thermal step. And we have done a complete turn. You can do it again, 4 steps, complete turn, etc. And so at the beginning, they were in the range of 1 turn within 1 hour or a couple of hours. And as it is usually the case, when you find something you believe very important, you can invest much more work in it, in the projects, and generalize the concept and improve the systems. And this is what they did. And now they are in the microseconds or even below time scale, and they have extremely efficient systems. Now let's go back to the rhotaxanes and the catenanes. And just to show you that we did what we had to do, having discovered a kind of a new principle for setting molecular molecules in motion, we decided to improve the systems, trying to speed up the motions, and we were successful. So from the original catenane published in 1994, we modified the structures. We tried to understand what were the limiting factors in terms of rate for these rearrangements. And we spent probably 12 to 15 years improving the systems. And in particular, for a pirouetting system of this type, we could reach the time scale of microseconds to milliseconds, which of course represents a very big improvement compared to seconds to minutes. So at the same time as our first catenane, which we call the pirouetting or oscillating catenane, the group of Fraser-Stoddart published a very beautiful piece of work. They made a molecular shuttle. And a molecular shuttle, I think, no need of explanations. As I already said before, you take the ring, the red ring, and you move it from the left to the right, and you go back to the left. And they call that molecular shuttle. The work was done by Ankel Kiefer and his professor at Florida University in Miami, and Fraser-Stoddart, and 2 postdocs, I think, Bissell and Cordova. Probably a Spanish guy, but I never met him, I don't know. So if you have the blue ring, which is here, which is very strongly electron deficient, it can interact with a strong electron donor, the green station, they call it. And now, if you completely cancel the attractive interaction between the blue box or the blue ring and the green electron donor, the ring will move to the right. And this is exactly what happens if you inject a positive charge in this diamine, aromatic diamine, by abstracting an electron. You generate a species which is now no more an electron donor, And this is why the ring will move to the right, so as to interact with a weak electron donor, this aromatic diether. And now you can go back by reducing back the radical cation, which was formed here, to the neutral form of the green station. So very nice work, which led to many very exciting studies and many publications. And I think I just selected one, which seems to be quite important. Fraser Stoddart and Jean-Habif and their many collaborators and co-workers were interested in a new field, trying to make, well, let's say that's very ambitious, molecular computers, or at least information storage devices based on molecules. And I think they succeeded, brilliantly. They were using a very long rataxane whose two ends were attached to nanoelectrodes, and they could write, read, erase by just applying some potential between the two nanoelectrodes, which were attached here and here, by moving the ring. And if you move the ring from the green station to the red station, you can write, let's say, 0 or 1, 1 or 0. Then you can read by measuring the electronic configuration of a bundle of such wires, so to say. And you can erase by applying a high redox potential, which will simply lead to everybody going back to the same state. So that was really a remarkable piece of work. In our group, of course, we have made many systems, which for some of them turned out to be exciting and efficient. And probably the most efficient system, or the most remarkable system, was made by Cello and Christian. I will mention their name immediately. It's a muscle. So at first, we have to remember how muscles work. And muscles work in a relatively simple fashion. Muscles work by gliding filaments. And probably many of you are aware of that. We have thin filaments, which are actin polymers, and thick filaments, which have an ATPase activity. And so if you use those thick filaments, which are at the center of this small elemental piece of muscle, let's say the sarcomere, by hydrolyzing ATP, in other words, by consuming energy, the filaments will glide. This sarcomere will contract, which means that you contract your muscle. And this is exactly what we had in mind, thanks to the work of Cello, Consuelo Jimenez, and Christian Dietrich Bochecker, who basically devoted all their energy and enthusiasm in the project. We could make a rataxane dimer which behaves as a muscle. I must apologize because it's a very complex system, and it will take at least 10 minutes of my time to enter all the details. So I'll just show you the principle. And the principle is shown here. If you have a rataxane dimer with two covalently independent units, the pale blue unit and the dark blue unit, you can observe that it glides using some chemical signal. You go from about 8 nanometers of length in the extended system to 6 nanometers when you contract. And so many people became interested in this system. And I think that nowadays there are several groups working on artificial muscles related to this principle. And in particular, some people could make fibers, micrometer size fibers, and related systems. And I believe it's a field which is promised to a bright future. So now let me finish up. This is the very last scientific example. Again, it is partly inspired by biology. In biology, there is a family of enzymes which is absolutely incredible, the chaperones or the chaperonins. And these enzymes are very, very complex. There are two subunits, GRO-ES and GRO-EL, and these subunits can form some kind of dimer of a complex. And there is a huge cavity which is generated when they assemble. And if you think of proteins, in particular enzymes, which are working in our cells, like all the proteins in the living world, after a while, they can denature. Another way to say it is that they get tired and you have to fix them, to repair them. And instead of throwing them away again, the cell tries to repair them, and there is this big enzyme. And this big enzyme will be able to encapsulate the denatured protein, do some massage on them to pull on parts, push on other parts, compress, elongate, and all that. And after some time, the protein will be completely repaired, fresh, and then it will be expelled and go back to work. And of course, to chemists, I think this is really fascinating to know that there are big molecular structures able to encapsulate smaller structures and modify their conformation by pulling on parts of them, pushing on other parts. And we thought that maybe it could be exciting to make a compressor or maybe a switchable receptor. So let me make it clear. The project is incredibly naive compared to biological systems such as the chaperonins, incredibly naive, almost stupid. But from a synthetic viewpoint, we thought it was very challenging. And so we started to think of it and we made the compound. So now let me spend a few minutes on that. That was the project, making a 4-rotaxane. Of course, there was a synthetic challenge. That was also one of the motivations for making such a molecule. We will have 2 dumbbells with 2 big stoppers here, 2 other big stoppers here. And we would have 2 Bis macrocycles, one here, another here, and 2 big plates, rigid plates at the centers of those 2 Bis macrocycles. I should say that the work was done by many people, including a Spanish postdoc, Felipe Reveriego, who turned out to be very efficient. So let's try to make that. You will be surprised, or I should say that the purpose was to incorporate a guest in between those 2 plates, and then to see whether we can compress by bringing those 2 plates to close proximity. And so, again, now this is the chemical drawing we have made. And this is not the structure, but let's say the drawing of the molecule. And it is exactly as what we had discussed before. We have the 2 dumbbells, we have the 2 Bis macrocycles, and the plates are zinc porphyrins, simply. to rigid plates. You know that porphyrins are very easy to work with. They are very stable and very rigid. And so we made a molecule which was, I should say, less difficult than expected because of symmetry. You know, when you have a very symmetrical molecule, in a way it is cheating. It is always much easier than when you have a totally asymmetric species. So we could make, if I remember well, 130 milligrams of this compound. And 130 milligrams, you can do everything. Now something which was unexpected, which is icing on the cake. We got the X-ray structure of the molecule, and this is the X-ray structure. You see that it's a relatively respectable compound. The molecular weight is close to that of a small protein. The size also is respectable, 50 angstroms, 45 angstroms. And we can easily see all the elements, the important elements. We have the dumbbells, white or pale gray with the 4 stoppers. We have the 2 porphyrins here with zinc ions and some space. The space is about 10 angstroms between those 2 plates. Now we can insert some small molecules like pyrazine, dapcode, diamines, various diamines, and we will insert them and see what happens if we can compress the system. So now there is a video here, an animation, and the animation is based on the X-ray structure. It's not science fiction, of course. And we will compress. And the compressed species is based on very detailed NMR studies. But again, this is not science fiction. We have all the data needed to allow this animation to be made. So the signal we send to the molecule will be removing the copper. We send we add small amounts of cyanide. And now you see that the 2 plates are becoming closer and closer to one another. And this is really the end form of the molecule. And if you had something in between those 2 plates, of course, it will be compressed and compressed. And at some stage, it will be expelled. Of course, you can remetallate, reintroduce your 4 coppers, and you regenerate the complexing ability of the system. You can reintroduce a small molecule between the 2 plates and go on. And so that was our approach of trying to make a compressor or a switchable receptor. Again, it is a complicated molecule, not so complicated, but relatively complex. And it is extremely naive, simplistic compared to any system taken from the biological world. So let me finish up by saying that there are nowadays many groups which have been interested in molecular machines, molecular compressors, molecules able to walk, and this is David Lee's work. The ribosome, which is the most complex biological molecular machine, has been mimicked by the group of David Lee. Of course, it's very naive compared to the functions of the ribosome, and many other examples. I had a small chapter on the applications, potential applications. I think I will partly skip that because of time, which is running very fast. Let me say that, in my opinion, the most important potential application is that on targeting malignant cells in the body, targeting the things in the body which are considered as aggressors and which have to be destroyed. And as you know, chemotherapy is still not very selective. And I believe that someday, either using nanoparticles, very sophisticated nanoparticles, which will behave as molecular machines or even molecular machines, we will be able to target cancer cells and try to destroy them. There is still some work to be done, no doubt. Let me finish up by saying that what I spoke about today was a collective piece of work, highly collective. We were lucky to have professors or professional researchers, and these are the people who very strongly contributed to this work. Christian Dietrich Buschecker, who is a research director, Jean-Claude Chambron, the same, Valérie Heiss, who is now a professor in our university, Jean-Marc Kern, who was a professor, who very sadly passed away, Jean-Paul Collin was a research director, and three younger members, many PhD students and postdocs. Let me mention more specifically the people who participated in the field of molecular machines, which I call inaction. I particularly insist on our Spanish connection, Diego Cardenas, who is in Madrid, Pablo Maria Consuelo Jimenez Chelo, who is responsible for the work on the muscles with Christian Dietrich, Maria Jesus Blanco Pilado, who everybody used to call Chus, who is now working in a big chemical company in the US, Felipe Reviriego, who is in Madrid. And the work I spoke about at the end, many people are on this slide. And finally, let me thank my university, the French CNRS, as Enrique told you, which is a bit similar to CSIC, but not exactly. The European communities. I also would like to thank my second university, Northwestern University, which I joined after the CNRS suggested me to retire. Well, in fact, it was more than suggesting me, it was forcing me. And Northwestern was very generous, thanks to Professor Stoddard, partly. They invited me to join, and I spent 3 years at Northwestern, commuting between Strasbourg and Evanston. It was a fantastic period of time. My former mentor, Jean-Marie Lehn, who is now a very close friend, Malcom Green, who passed away 1.5 years ago, very sadly, a great organometallic chemist, who was my postdoc supervisor, 2 teachers, my family, and my 2 good friends, Professor Stoddard and Ben Feringa. And finally, let me thank you for your attention. Thank you.
Okay. Thank you very much for this superb lecture. Very, very amazing, fascinating.
Thank you.
I heard you for the first time in 1987 in Florence, and you spoke about the first 2 catenanes, and already was the audience was very interested. And I'm sure there will be plenty of questions. I think Jose will be up. Any questions, comments? You can write it on the chat or raise your hands.
Ya todos tenéis habilitado el chat, ya tenéis habilitada la cámara, ya tenéis habilitado el micrófono, si queréis hacer alguna pregunta.
Okay.
Pablo, I think.
Just to thank you for this wonderful conference. I know very well your work, so and I'm inspired, and it was like the lottery for me to work with you. So.
But you are also doing very nice work yourself now in this field.
Okay, good. In the meantime, as people, when you use this copper 2, copper 1, how many cycles can you perform with?
Yeah, we never detected any fatigue. It's totally stable, perfectly stable. The reason why, in a way, is that there is no real chemical reaction.
Yeah,
you oxidize something, it's a redox process, and then you have two rings which rearrange, and that's all.
And that's also so you can
never observe it.
You can inject electrons and without
You inject the electron, you take it back, and nothing bad can happen in a way.
So it's a clean input, electrons.
Yeah, I think it is different, with purely organic systems. I mean, the work of Fraser Stoddart is absolutely fantastic. But in this case, you generate organic radicals.
Yeah.
If you reduce the blue box, you generate a radical. Maybe it's going to be very stable, but probably less than copper 1 and copper 2. Copper 1 and copper 2.
Yeah.
Except if you are able to do a nuclear reaction, nothing is going to happen.
Okay.
Yeah. Thank you. Thank you.
No questions yet. And in the compressor, the guest between the porphyrins, which was the guest? Which cyanide? The guest.
No, small molecule with two nitrogen atoms, like DAPCO or 4,4' bipyridine, and each nitrogen atom is connected to the zinc center of the porphyrins.
Okay.
I don't know if you see that. DAPCO, it's you have two nitrogens, and 4,4' bipyridine. You can take a diamine, small diamine with two nitrogen atoms.
Two nitrogen.
You have to
It's adamantine also.
Yeah, sure. Yeah. And so
Okay.
there
And
are
the metal ion is zinc too, no? The metal ion in the porphyrin.
Yeah, it's zinc.
Yeah. I think there's question. Yes.
Oh, don't worry, it was not a lecture for generating questions. It was a general.
Okay, there is a student that is asking that he doesn't know how to ask in English, but I will try to translate it. He's asking you, he hasn't understood why the molecule, I think that is the molecular light-induced motor, about that. He hasn't understand that why the double bond breaks and by the action of the UV light, and also recomposes afterwards, and that induced a conformation change of the molecule. That's
so I share my screen again. Yeah. Can you see that I shared my screen?
Yes, we are looking at it.
So here you have a double bond, and this double bond is not so happy because you have many groups. You have, of course, 4 groups here, and those groups are sterically hindering. They are compressing each other. There is some steric hindrance. But a double bond normally cannot rotate. If you remember your organic chemistry lessons, a double bond is rigid. It doesn't rotate. Now, if you shine light, you generate what is called an excited state. And this excited state, you can look at it as if it were a diradical. You have a radical here, you have a radical here, and a single bond. I think this is the best way to explain. If you have a single bond, now a radical here and a radical here, a single bond, of course, rotation can be free. And now you start to rotate because it's a single bond, and the system will prefer to have a conformation which diminishes the steric hindrance between the various groups in the vicinity of the double bond. Is it clear? I'm not sure it's clear.
There's another question by.
Okay, the student says that he understands now, and now other student is asking. Celia Garcia asks, which was the most difficult catenane that you have synthesized, and how did you face it?
Yeah, I mean, we made the catenane with seven rings. 7. So there was a huge ring in the middle, which was 140 atoms, and 6 smaller rings around it. And the people used to call that a necklace, you know, like something you put for ladies mostly around your neck with small beads, you know, small beads. And but it was not the most difficult. The most difficult to make was Solomon link, which is a doubly interlocking catenane. It's difficult to show you. So you have this, and you turn twice for the second. Okay. I think I can show you again. You know, let me show you. Yeah, it's here. And I will share my screen, okay. So do you see it?
No.
Do you see my screen?
No.
Yes?
No.
No, not now.
No.
No, we are not able to take a look at your screen.
Yeah.
Now.
No,
okay.
Yeah,
perfect.
Yeah. Sorry. And so this is Solomon catenane. It is called Solomon catenane. If you look, you know, the rings cannot be flat. There is some distortion. Yeah. And you need to have 4 crossings. But the most difficult species in terms of topology was the trefoil. It was terrible.
Okay.
Okay.
Okay. I think the students are coming up with new questions.
Okay, good.
Through the chat, David De Hoz is asking, how do you insert or subtract electrons selectively in certain groups in big molecules? How do you do that?
Yeah. How can we introduce electrons at the right place? Yeah, this is the
yes, that's the question.
So this is based on electrochemistry. So I will share again, you know, I will share my screen again. So if you, what should we take? And let me take the first molecular machine here. So do you see the screen?
Yes, yes, we can see it.
So in this case, there is no doubt that if you have an electron here, the electron will go where the you have the best electron acceptor. If you want to inject an electron somewhere here, you have to have a place for the electron to go and stop. And the only place where the electron can go here is the copper 2 in this case. Copper 2 plus, it's a good electron acceptor. But you cannot put it in the terpyridine group, as Enrique would fully agree, I'm sure. You cannot easily put it in the terpyridine part, So the electron has to go where it is the easiest to inject an electron. And the easiest way is to go to the copper 2 center, just to make it simple.
Yeah.
Does it answer the student's question?
Okay, thank you, Jean-Pierre. Another student, Adriana Ramirez, is asking that she doesn't understand how the catenane move along the shuttle compounds. Why they move? The ring along the
proton.
The catenane. The driving force of that movement.
Is it for this compound here?
It should be the Stoddard compound, probably the one that's protonated.
I think
protonated.
that.
Yeah. So do you see it?
Yeah, that's protons.
Yeah.
Yes.
You see. So it's very simple. It moves because it is unstable. I think this is the principle of all the molecular machines, basically in the world. Even in biology. If you have a molecule with a certain shape or a certain conformation, and if you send a perturbation, you send a signal to the molecule, and you make it feel very bad. You make it very unstable. What will happen? The molecule is not stupid. It will not stay very, very unhappy, very unstable. It will try to relax. This is the way physicists say. There is a relaxation process. And the relaxation process implies motion. It will move and find a better or acceptable situation, which will make it feel much happier. Does it answer the question? I hope it does.
That the question is perfectly answered. Two more questions there in the chat. David Garcia tells you that when you talk about rotary motor, the video show how the Atp Synthase rotates over and over again. It was a quite impressive video. But when you twist two ropes, there is one point where you can't twist it more. In this compound, happens the same?
Yeah. No, you can twist it. Let's go back to this video. So you see the molecule, don't you? No?
Yes. Yes,
yeah. You see the molecule. Look, here, if you rotate, I'm not using to use the movie, but if you rotate here, you can rotate as many times as you like. It's no problem. Think of ethane. Ethane, you have CH3, CH3, and a single bond between the two carbon atoms. Okay? Ethane. Yes? Does speak to you, ethane. Sure. And now you can rotate one CH3 as much as you like, and the other will not care. So you can rotate, as much as you like, and there is no barrier in a way, there is no strain. You can rotate as much as you like, what you need is some energy to pass the barrier, because you have steric inputs. But otherwise, you can do it as many times as you like. And if I may just go on this particular system, because there is some beautiful work. It was done in Strasbourg, by a young group, just name. So they used more or less the same system, but they attached it. So they attached long chain between, say, this point and this other point, a very long chain. And now they try to rotate. And if you have a long chain, of course, it's totally different. It is like having a string, and then you start to make a knot, the string, and you can only rotate 1, 2, or 3 meters, never more. And so that was also a knot.
Yeah.
So did it answer the question?
Yes. Yeah, sure.
Yeah, sure.
Okay.
Now there's a question by another student, and he says that is copper the most stable and cheapest option to form these compounds, or there are more metal ions that could be better, but they are way too expensive, just like ruthenium? This is the question of Juan Briones.
This is a very good question. I mean, all the questions are good, by the way. There are bad answers, sometimes. But this is a very good point. Yeah, we have been working on ruthenium, and we made some ruthenium-containing catenanes, and we could also set them in motion. And the motion was totally different. The principle was totally different, because with ruthenium complexes, when you shine light, when you photo-excite these complexes, in some cases, you can expel a ligand. You can completely rearrange the system. And this is what we did. We had a catenane which we could completely rearrange using light by photo-exciting the system. But of course, these systems are much more expensive. They are fun. They are very different from the copper-containing catenanes, but very, very expensive. And so we didn't work so much in this field. We spent maybe 10 years or so with ruthenium and rhodium complexes. But copper is still our favorite metal.
And Jose?
And think that there is a final question about other student, Ainhoa Martin, that says that you're talking about the future application for the treatment of cancer. The objective is searching for a way of marking the malignant cells so that the body will be able to destroy them itself, or the synthesis of new drugs, either to a way to marking the malignant cells or synthesis of new drugs.
No, it's also an interesting point. Some people try to make new therapeutic molecules, new drugs, against cancer. It's a field. It's a huge field in itself. There are nowadays thousands and thousands of new drugs. Some of them being very, very efficient, more and more efficient. But it's not our field. I think our field is some means to, let's say, carry the drugs, to really have some kind of vehicle carrying the drug close to the cancer cell and then injecting the drug into the cancer cell. It's very different. So what the people will do in the field of molecular machines, they will use drugs which are known, which are very well established, but they will carry them. They will have some kind of container, they will put some some of these molecules in a container, close the container, and have the container traveling in the body, and then injecting the drug in the cancer cell. If I may say, there is something which was done a couple of years ago, which is fantastic. This is the work of James Tour and his group in the US. They have used Ben Feringa's work. They have used a rotary motor, and they attached some groups on the Ben Feringa's system. And when they shine light, you know, the rotary motor can drill a hole. So it is very close to the cell. They use a cell, and they drill. I don't know if you know the words, you know, to make a hole, you know, a drill. They make a hole, and they can inject something in the cell.
In Spanish, we say taladradora.
Yeah, absolutely amazing.
Cadena, as you asked before, chain.
Yeah.
In Spain, we say cadena, and chain mail, we say cota de malla.
Okay,
So,
cota de malla. Yeah.
Maybe not a difficult word.
No.
So there are questions, but I think it's time to close the session and to thank the students for their participation, and Jean-Pierre for this fantastic lecture. And I hope to see you soon here in Valencia, probably, and face to face. And
yes,
and thank you very much indeed. It was very, very interesting for all of us. Thank you, Jean-Pierre. I don't know.
Thank you to Jose, to you, and to the students.
There are many students that are
saying thank you,
saying that they enjoy very much the lecture. Okay, thank you.
Good to know. Okay.
Thank you very much.
Bye-bye then.
Bye-bye.
Bye.
Ciao.
Bye.
See you.